Assalamu alaikum, dear students. This is me, Atiq Ahmad. Today we are going to study the urine formation process. During the previous lecture, we have started the broad topic, that is the excretory function of nephron. We have divided this broad topic into two parts. In the first part, we have studied how the blood is supplied to the kidney, how it is distributed inside the kidney, and then how the blood leaves the kidney. So we will have a brief overview of the previous lecture, and then we will proceed towards the today's lecture, that is the urine formation. So the blood is supplied to the kidney through the renal artery. The renal artery divides and redivides and form fine branches and ultimately form finer arteries, which are known as cortical radial arteries or peritubular arteries. Then these arteries further divide and redivide to form afferent arteriole, which supply blood to the glomerulus. The glomerulus is actually formed from the afferent arteriole itself. Afferent arteriole divide and subdivide into fine capillaries, and their tuft of capillary is known as glomerulus. Whenever the filtration occurs inside the glomerulus, so the blood leaves the glomerulus through an arteriole which is known as afferent arteriole. The afferent arteriole divides further and form peritubular capillaries, which surrounds the proximal convoluted tubule and the distal convoluted tubule. The specialized area of these peritubular capillaries is known as vasa recta, which descend down and then ascend upward along the loop of Henle, and where the reabsorption process takes place. After that, the peritubular capillaries, they combine together to form the interlobular veins. Then the interlobular veins drains into the renal vein. The renal vein then drain into the inferior vena cava. The inferior vena cava carry the blood to the heart. So this is all about previous lecture. Today we will move toward the second part of this lecture, that is the urine formation. The urine formation basically involves three processes: ultrafiltration, selective reabsorption, and tubular secretion. Ultrafiltration, glomerular filtration, or pressure filtration. These are the three names of this process. It is known as ultrafiltration because this is a high efficient filtration. It is known as glomerular filtration because this process takes place inside the glomerulus. It is known as pressure filtration because this process takes place under high pressure. So this process takes place in renal corpuscle under high pressure, known as hydrostatic pressure. The process takes place in renal corpuscle. Just recall, renal corpuscle consists of two parts, the glomerulus and Bowman capsule. So this is the actual filtration unit where the filtration takes place under high blood pressure. Now, how this high blood pressure is created? So remember, this high blood pressure basically originated from the contraction of heart, because the renal artery supply the blood to the kidneys. So the high pressure is felt inside the renal artery, and that is then transmitted to the arterioles, and then the afferent arterioles, and then the tuft of capillaries, the glomerulus. So this is the high pressure which originated basically from the heart. Now another point is that how this pressure becomes so high, that is that the diameter of efferent arteriole which conducting outward, that is half as compared to the efferent arteriole which conducting inwards. So the efferent arteriole which is moving inside the glomerulus, that has diameter double than that of the efferent which is leaving the glomerulus. So when the diameter of the efferent arteriole is higher, so it means that the blood pressure will be high, and that blood pressure is transmitted inside the capillaries of the glomerulus. So in this way, there are two basic reasons due to which the high pressure is created. One, the origin, that the contraction of the heart basically produce so much high pressure which is felt inside the renal artery, and that renal artery enters into the kidney, and then it is subdivided into the cortical radial arteries or interlobular arteries, in other words, and then those interlobular arteries form the efferent arterioles, and then the efferent arterioles divide and subdivides to form the capillaries network, the tuft of capillaries which is known as glomerulus. So first, at the origin the high pressure is created. Secondly, the efferent arteriole which is entering into the glomerulus, that has high diameter as compared to the one which is leaving the glomerulus, the efferent one. So because of these two reasons, high pressure is created, and due to this high pressure the blood is filtered. Due to the high pressure, the blood is filtered from the capillaries, and the filtrate is collected inside the Bowman capsule. So due to the such high pressure, water and small solute molecules are filtered out of glomerular capillaries and are collected into the Bowman capsule. Now the smaller molecules basically pass through the wall of capillaries, and then the double wall structure of the Bowman capsule, and the glomerular filtrate is formed. The larger molecules like proteins as well as RBCs, WBCs, platelets, etc., these large molecules are left behind inside the blood because they cannot pass through the small pores of the walls of capillaries, as well as of the basement membrane, as well as of the walls of the Bowman capsule. So the large molecules left behind, and the small molecules, water molecules, as well as the solute molecules, the various amino acids, the sugars, the vitamins, etc., etc., they are filtered out and are collected in the Bowman capsule. The filtered fluid in the Bowman capsule is called glomerular filtrate because it is obtained as a result of the filtration of the glomerulus, the filtration which occurred as a result of high pressure. That is why that is known as glomerular filtrate. It has chemical composition similar to that of blood plasma because it lacks the blood cells as well as the high molecular weight protein, and it contains the small molecules, water and other solutes just like the blood plasma. Now the second step, that is the selective reabsorption or tubular reabsorption. In the first process, the ultrafiltration occurs and the glomerular filtrate is formed, which have small molecules, water plus solutes. That solute consists of amino acid, glucose, fatty acids, vitamins and other small molecules. Now the ultrafiltration produces around 125 ml of glomerular filtrate per minute, which is equivalent to 180 liters per day. But In fact, this whole filtrate will not move in the urine. 124 ml of this filtered glomerular filtrate, it will be reabsorbed. So just 1 ml as a average, 1 ml will pass per minute. So actually the 125 ml of filtrate produced per minute, average 124 ml is reabsorbed, because the reabsorption process is very selective and useful substances for the body are reabsorbed. This is known as selective reabsorption, because selectively those substances which are important for the body will be reabsorbed. So glucose, amino acids, vitamins, inorganic salts and some water will be reabsorbed, and only the nitrogenous phase and the harmful substances and the harmful toxins and the extra materials that will be retained inside the glomerular filtrate, inside the renal tubules. So the reabsorption will occur where? This is the question. The reabsorption will occur in the renal tubules, in the proximal part of the renal tubules, in the loop of Henle, and in the distal convoluted tubule, and in a very least amount in the collecting ducts. But most of the reabsorption will take place inside the proximal, loop of Henle and distal convoluted tubules. Now we have studied the 2 steps. Just connect the 2 steps, then we will proceed toward the 3rd step. In the first step, the ultrafiltration occur under high pressure, and as a result small molecules will be filtered out and will move from the glomerulus into the Bowman capsule, and the glomerular filtrate is formed. Now the small molecule means water molecules and small solute molecules, glucose, amino acid, fatty acid, etc., etc. In the reabsorption process, the important small molecules will be reabsorbed, as well as water will be also reabsorbed. The small molecules, amino acid, fatty acids, inorganic salts, some water, these all are important for the body, so they will be reabsorbed while the movement of the glomerular filtrate inside the proximal tubule, the distal tubule and the loop of Henle. Now the 3rd step, the tubular secretion. This tubular secretion process is basically to maintain the pH of the urine which is formed. It is a process by which certain substances, ammonium and hydrogen ion, are secreted mainly by the tubular epithelial cells of the loop of Henle and to the lumen of the tubules. So whenever the glomerular filtrate, now later we will call it urine, so whenever it passes through the loop of Henle, so the walls of these tubules of this loop of Henle, they release ammonium and hydrogen ions to maintain the pH of this glomerular filtrate, or in other words it is now urine. So this pH, the normal pH range is 4.8 to 7.5. So this will be maintained on, and for that purpose the hydrogen ions will be released to make the urine a bit acidic, because hydrogen ions cause a bit acidity. So similarly ammonium ions will be released. So as a result the pH will be maintained within this range, 4.8 to 7.5. So slightly acidic, this is slightly acidic, this lower range, and this is in the neutral range. So this range is between the slightly acidic and the neutral range. So this range will be maintained through the tubular secretion, the secretion of various ions, ammonium and hydrogen ions. Now the urine concentration process. Reabsorption of water takes place along the whole length of the nephron. Yes, we have discussed. But the formation of more concentrated urine is dependent on the reabsorption of water from the loop of Henle and collecting ducts. This is achieved through a process which is known as counter-current multiplier mechanism or counter-current multiplier system. Now first understand the terminology, so then you will better understand the concept. Counter-current means the movement of materials through energy both ways, in opposite direction, from one way as well as from the other way. So this is counter-current, the movement of material through the use of energy and in opposite direction. This is counter-current. So this counter-current process will take place and it will multiply the process. Multiply the process of what? The process of reabsorption, because in this case the concentrated urine will be formed. So what happens inside? Due to the counter-current, filtrate moving in the limbs of loop of Henle and the blood moving in the capillaries of vasa recta, around 99.5% of water is reabsorbed. And how it is reabsorbed? As the fluid travels up the ascending limb, sodium chloride is transported actively out of the limb into the surrounding area. So diagrammatically we will show it here that whenever the fluid travels up the ascending limb, this is the loop of Henle. So whenever the urine, now we will call it urine, or in other words it is glomerular filtrate, but later on it will become finally urine. So whenever it moves through this ascending limb, so what happens? Through the use of energy, this is ectotransport, sodium chloride ion, NaCl ions are transported from this lumen, from this inside, into this space, into this space. So as the fluid travels up the ascending limb, sodium chloride is transported actively out of the limb into the surrounding area, into that space which we have seen diagrammatically. This movement is controlled by adrenal cortical hormone, aldosterone. Now this message is transmitted for the concentrated urine formation by two hormones. Two hormones are basically involved. One is this adrenal cortical hormone and the other is ADH, antidiuretic hormone. Because whenever the water supply to the body is low, there is less water content inside the body, so concentrated urine should be formed, and for that concentrated urine formation, message is transmitted from the brain to the posterior pituitary gland as well as to the adrenal gland. So the adrenal gland will release the adrenal cortical hormone, the posterior pituitary will release the antidiuretic hormone. These two hormones will travel through the blood and will reach to the loop of Henle and the other renal tubules, the proximal and distal tubules. And under this signal, the sodium chloride ions are actively transported from the ascending limb into the surrounding space, into the surrounding area. Now what happens as a result of this transport? This causes an increase in the concentration of water in the filtrate because the solute is transported out of it. So as a result, the water concentration will become higher and the solute concentration will become lower. Now in the space where the sodium chloride are released, there the solute concentration will increase. So as a result, the solvent concentration will decrease, the water concentration will decrease, while from the inside of the ascending limb, from where the sodium chloride is transported into this space, there the solute will become less and the solvent will become higher. The water will become higher. So what will happen as a result? As a result, water passes out of descending limb by osmosis. Because in the space there is less water and more solute. Now look at here. From this ascending limb, the sodium chloride are transported here, as we have described. So here in between this space, in this space, the sodium chloride concentration become higher and the solvent concentration will become, the water concentration will become lower. While inside, the opposite case will occur. The solute concentration will be lower and the solvent concentration will be higher. Now what will happen? Now here the process is different. Now from here, the solvent, the water will move to this interstitial space. Because here the water is less and inside this limb, the descending limb, the water concentration is higher. So the water will move through osmosis from the higher concentration to the lower concentration here into this space. Now what ultimately will happen? Ultimately this water will be reabsorbed by these capillaries, blood capillaries, which are surrounding this loop of Henle. So as a result, the water will be retained and that water will flow inside these capillaries into the renal vein and then in the inferior vena cava, and in this way the water will be retained within the body and the urine will be concentrated. So this is all about today's lecture. Now look at this diagram. This is the efferent arteriole. This efferent arteriole enters into this Bowman capsule. Through this efferent arteriole, the blood is supplied to these tough top capillaries, the glomerulus. Now this blood is supplied through the efferent arteriole through high pressure. Now there are two reasons how the high pressure is created. First, the heart which pumps the blood, from there the pressure is created. Due to the contraction of heart, the blood is pushed inside the arteries. And remember and recall that the renal artery enters into the kidney. That renal artery then divides and redivides to form cortical radial arteries, and then it further divides to form these efferent arterioles. So this is that pressure which is basically originated inside the heart, and that is felt here. This is one reason that the high pressure blood is transported here inside the glomerulus. The second reason of the high blood pressure is that the diameter of this efferent arteriole, it is double than this efferent arteriole which is leaving the glomerulus. So due to this high diameter, also the pressure is further increased, and partly it is due to the low diameter of the efferent arteriole. So the high diameter of the efferent arteriole, which is conducting the blood inside the glomerulus, and the low diameter of the efferent arteriole, this also contribute to the high pressure of the blood inside the glomerulus. And this high pressure is basically required for the filtration process, because this is high pressure filtration, and due to high pressure the efficient filtration will occur. So after the filtration, the glomerular filtrate is formed inside this greenish Bowman capsule. This will move inside this proximal tubule, then it will move into this loop of Henle, then it will move up into the distal convoluted tubule, then into the collecting duct, the collecting tubule, and then to the pelvis, and from there through the ureter, and then it will be stored in the bladder, and then it will be released through urethra. So during the first step, the pressure filtration occur, the ultra filtration occur. This is the pressure filtration or the ultra filtration under high pressure. Then the selective reabsorption process takes place. The selective reabsorption takes place inside these proximal convoluted tubules and the distal convoluted tubule. And the third step is the tubular secretion process. So these tubules of loop of Henle, these renal tubules, the walls of these renal tubules, basically secrete certain ions, ammonium ions and hydrogen ions, to maintain the pH of this glomerular filtrate, or in other words, it is urine now. So its pH must be maintained from 4.8 up to 7.2 or 7.3, in this range. So this range is basically maintained through the tubular secretion, through release of ammonium and hydrogen ions, to maintain the pH within this range. In the counter current multiplier mechanism, what happens basically, this mechanism is applied whenever the blood supply, whenever the water supply to the body is low, the body contain less water. So in that case, what will happen? Counter current multiplier system will play its role. What is this system basically? First, you people have to understand the counter current. Counter current is basically the flow of material from both sides in opposite direction through the use of energy. This process basically takes place, the counter current mechanism or process takes place inside this loop of Henle. What happens basically, whenever there is less availability of water inside the body, there is less water content. So in this case, the kidney should form concentrated urine and more water should be reabsorbed, and that reabsorption process is achieved through the counter current multiplier mechanism. Counter current multiplier mechanism. This mechanism will further multiply the reabsorption of water, which is already performed by these proximal and distal convoluted tubule, but this mechanism will enhance that process, will multiply that process. That's why it is known as counter current multiplier system. What happens in this process, that in this ascending limb, whenever the glomerular filtrate move inside this limb, so what happens, through the use of energy, sodium chloride ions are expelled out from this filtrate inside this space. What happens basically, the solute molecule, the sodium chloride, is removed from here. So here the water concentration will increase, the solvent will increase, while here the solute concentration will increase and the solvent will decrease, the water will decrease. So as a result, what happened here, the process is normal. So what will happen in this descending limb? In this descending limb, the water concentration is higher and the solute concentration is lower. So from here, the water will move through the process of osmosis from this inside into this interstitial space, into this space. So what will happen? From here also the water is released. From here the water is also released. So ultimately the these vasa recta, these are vasa recta, the specialized area of the peritubular capillaries. This is called as vasa recta, this specialized area which is around the loop of Henle. It will absorb that water from this space, and as a result that water will flow inside the blood, and inside the blood it will be retained within the body, and here the concentrated urine will be released from the body through the collecting duct and then through the ureter and then passing through the urethra after brief storage 2 to 3 hours in the urinary bladder. So this is the whole process which we have described here diagrammatically. See you next time with new lecture. Take care. Allah Hafiz.